Off California's coast on the sun-browned Channel Islands, a creature the size of a small cat evolved in splendid isolation. Eurasian littoralis, the island fox. It developed on 6 islands: San Miguel, Santa Rosa, Santa Cruz, Santa Catalina, San Nicolas, and Santa Barbara. For millennia, the Chumash and other native peoples managed these oak-dotted coastal scrub landscapes. European contact and 19th century ranching brought sheep, cattle, and feral pigs, altered vegetation, and layered the islands with human disturbance. The islands sit 20 to 85 miles off the Southern California coast and were partly protected when Channel Islands National Park was established in 1980, yet their ecological webs remained compact and precarious. Island foxes were the dominant native mesopredator, quietly shaping seabird colonies, insect populations, and seed dispersal. Before the 1990s, biologists described stable or locally abundant fox populations across the islands and recognized 6 named subspecies, each genetically distinct and geographically restricted. That apparent equilibrium, a triumph of dwarfing and endemism, masked deep vulnerabilities, tiny population sizes, restricted ranges, and new pressures from non-native mammals and habitat change. Those factors primed the archipelago for a sudden collapse beginning in the mid-1990s and prompted an unprecedented conservation response. Scientists such as Dennis Garcelon would soon step in to intervene. In the mid-1990s, that gilded equilibrium shattered. Between 1994 and 1999, biologists surveying Channel Islands wildlife witnessed catastrophic crashes on Santa Cruz, Santa Rosa, and San Miguel. Field teams from Channel Islands National Park and the Institute for Wildlife Studies recorded declines exceeding 90% on some islands, and populations were driven to the brink on others, a pace and scale that stunned long-term monitors. Rangers and researchers, including Dennis Garcelon, moved from routine monitoring into crisis mode as carcasses and missing radio-collared animals accumulated on bronze-scarred beaches and windswept ridgelines. Initial counts in 1996 and 1997 showed numbers collapsing far beyond normal fluctuation, and by the late 1990s, island-wide surveys found only a handful of foxes remaining in areas that had once been full of them. Necropsies and field observations offered grim clues. Trauma consistent with raptor predation, owl and raptor pellets, and a shifting predator landscape that biologists linked to expanding golden eagle activity, Aquila chrysaetos, on islands that had previously held only the endemic, much smaller raptor assemblage. Managers also launched systematic pathogen testing, screening for canine distemper virus and canine parvovirus to determine whether infectious disease was a contributor. Every day brought new data and new urgency. Maybe clicking subscribe would have saved a fox or two. Radio telemetry teams intensified tracking of survivors, NPS and U.S. Fish and Wildlife Service staff convened emergency meetings, and the Santa Barbara Zoo and other institutions began discussing captive holding protocols. Local ranching histories and the presence of feral pigs, introduced in the 19th century, were quickly implicated as an ecological link drawing eagles ashore. By the close of 1998, the crisis led to one unavoidable conclusion. Rapid, coordinated intervention was necessary, and the stage was set for the focused recovery campaigns that followed. Between 1996 and 1999, field teams shifted from alarm to methodical forensics, connecting scattered losses into a coherent, 2-part explanation. Necropsies by Channel Islands National Park veterinarians and Institute for Wildlife Studies biologists revealed a repeatable trauma pattern: crushed skulls, talon punctures, and disarticulated carcasses consistent with large raptor attacks rather than mammalian scavenging. Eagle pellets collected in 1997 and 1998 on Santa Cruz and Santa Rosa contained fox fur and small bones. Technicians at the Santa Barbara Zoo and university labs cataloged that material and matched it to the island fox, Eurasian littoralis. Radio telemetry proved decisive. Radio collars on surviving foxes sent abrupt mortality signals. Crews found collars near eagle roosts, and staff followed fresh sign back to kill sites. Observers photographed and videotaped golden eagles, Aquila chrysaetos, coursing over ridgelines and bringing pig carrion from feral herds onto the islands, confirming a behavioral link. Historical context mattered. Feral pigs, introduced in the 19th century and concentrated on Santa Cruz and Santa Rosa, created an abundant food source that drew mainland golden eagles ashore and altered a decades-old predator regime. At the same time, laboratory teams began running serology, PCR, and virus isolation tests for canine distemper virus, spelled out as CDV, and canine parvovirus, spelled out as CPV, starting in 1997. Pathogen screening showed disease was a concurrent stressor, with evidence of parvovirus exposure and at least localized distemper infection in the late 1990s. Taken together, the telemetry, pellet analysis, necropsies, and lab diagnostics turned a mystery into a clear explanation. Golden eagle predation, facilitated by introduced pigs and ecosystem change, and concurrent infectious disease were the twin drivers of the mid-1990s island fox collapse. That conclusion set the urgent agenda for the rescue operations that followed. Beginning in 1998, an urgent all-hands campaign coalesced under Channel Islands National Park, the U.S. Fish and Wildlife Service, the Institute for Wildlife Studies, and partners including the Santa Barbara Zoo, San Diego Zoo, and Los Angeles Zoo to stop the collapse of fox populations on Santa Cruz, Santa Rosa, and San Miguel. Teams moved fast. Captive breeding protocols were written, founder males and females were trapped in late 1998 and 1999, transported under veterinary oversight to zoo facilities, routinely vaccinated for canine distemper and parvovirus, and hand-reared when necessary to bolster survival. Dennis Garcelon and field biologists coordinated radio telemetry and genetic management to avoid inbreeding in the temporary holding pens. Concurrently, wildlife managers targeted golden eagles, Aquila crisatos. Beginning in 1999, Cruz used baited traps and telemetry to capture and remove island hunting eagles, relocating some to mainland facilities and removing others from the islands to stop ongoing predation. Feral pig control on Santa Cruz and Santa Rosa reduced an attractant that had drawn eagles ashore, using removal and containment measures led by NPS staff. Releases began as staged trials in 1999 to 2002. Vaccinated radio-collared foxes were soft-released into secured enclosures and monitored for survival, disease, and reproduction. The work faced stubborn logistical hurdles, limited founder numbers, transport stress, keeping wild behaviors in captive-reared foxes, and the complexity of testing every animal for pathogens and setbacks, including early post-release mortalities and public outcry over eagle removals. Ethical controversies simmered. Animal welfare advocates challenged eagle relocation and lethal options. Zoos debated hand-rearing versus fostering, and managers balanced rapid action against long-term genetic stewardship. Those tensions shaped every decision and pushed the program into a decisive reintroduction phase. The release of captive-bred, vaccinated, radio-collared island foxes from 1999 through 2002, and in the years that followed, became the decisive turning point. On Santa Cruz, Santa Rosa, and San Miguel, teams saw short-term survival improve, and foxes begin breeding in successive seasons. By 2004 through 2007, soft-released cohorts were appearing annually and integrating with remnant wild foxes. Intensive monitoring by Channel Islands National Park, the U.S. Fish and Wildlife Service, the Institute for Wildlife Studies, and zoo partners documented rising survival, sustained recruitment, and expanding territory occupancy. Removing golden eagles and eradicating feral pigs eliminated the main attractant and immediate predator pressure, while rigorous vaccination protocols limited canine distemper and parvovirus. Genetic management prevented inbreeding through careful founder selection and cross-island translocations. Field counts and island-wide surveys showed marked population rebounds. Localized extirpations were reversed and former strongholds repopulated. The program's cumulative success led to formal recognition. On October 4, 2016, the U.S. Fish and Wildlife Service removed the island fox from the Endangered Species Act, signaling legal recovery. By delisting, surveys confirmed restored ecological roles and managers shifted focus to long-term monitoring. That vindication closed the emergency chapter and opened an era of vigilant stewardship across the archipelago. The island fox saga began with a 1998 emergency that launched captive breeding and coordinated removal and reintroduction efforts. From that crisis to the U.S. Fish and Wildlife Service formally delisting the fox in 2016, the story offers a clear, pragmatic blueprint. Rapid, science-driven, multi-agency action by Channel Islands National Park, U.S. Fish and Wildlife Service, the Institute for Wildlife Studies, and zoo partners, paired with invasive species control, can halt steep declines and restore ecological balance. The program now stands as a model for island restoration, showing how targeted predator management, feral pig eradication, and strict vaccination protocols can avert collapse. But the victory is fragile. Long-term stability depends on continuous monitoring, sentinel disease surveillance for parvovirus and distemper, and readiness to respond if predator pressure returns. The campaign also forces uncomfortable ethical questions about removing predators and balancing animal welfare with ecosystem health. What hard choices will future recoveries demand?